Petrochemical refineries and automotive plants rely on unbroken production lines. Raw materials enter one end and finished products emerge from the other, continuously. The pharmaceutical industry, however, has historically operated differently. While small molecule drugs like antibiotics and vitamins have successfully shifted to continuous manufacturing, the production of large molecule biologics, like monoclonal antibodies, remains tied to discrete, step-by-step batch processes. Monoclonal antibodies are structurally intricate proteins produced by living cells. This introduces immense biological variability. The raw materials are sensitive, and the living systems that produce them are highly dynamic, making a steady, continuous output difficult to stabilize. Furthermore, maintaining an uninterrupted flow of live biological material requires constant measurement. Historically, the industry lacked the real-time analytical systems needed to monitor and control these biological fluxes instantly. Implementing a continuous bioprocess is not solved by simply stringing batch tanks together with hoses. The entire pipeline, from the initial genetic clone to the final drug formulation, must be completely re-architected. The baseline rules for this new factory floor originate from the FDA's Quality by Design, or QBD initiative. QBD requires mapping the production process so thoroughly that product quality is engineered directly into the system parameters from the start. We do not test the product for quality at the end of the line. The design of the line itself ensures the quality. To execute this, the system relies on Process Analytical Technology, or PAT. This serves as the real-time sensory network. Instead of pausing production to run slow, labor-intensive offline assays, the PAT framework embeds the sensors directly into the flow. It monitors critical attributes inline, online, and at-line. Pairing a robust process design with an embedded real-time sensory network allows for the removal of human intervention. This combination is the foundation for a fully automated, continuous pipeline. A continuous pipeline begins at cell line development, long before a bioreactor is ever turned on. Because continuous perfusion runs can last weeks or months, the selected cellular clone must possess absolute genetic and phenotypic stability. If the cell line mutates or degrades, the entire downstream process fails. Once stable cells are introduced to the upstream perfusion bioreactor, the mechanics diverge from a traditional batch culture. In an alternating tangential flow system, a filtration mechanism continuously separates viable cells from the nutrient fluid. The cells are retained while the product-rich permeate is pushed out. Within this reactor, continuous data feedback loops regulate dissolved oxygen, pH, and carbon dioxide levels to keep the cells highly productive. As the culture scales, cell densities can reach an extreme 200 million cells per milliliter. To prevent the system from clogging, a capacitance probe tracks the density and triggers an automated cell bleed, skimming off excess biomass to lock the reactor into a steady state. Simultaneously, automated aseptic sampling systems use uniflow hollow fibers to extract minute amounts of cell-free media. This feeds into offline spectroscopy to track metabolite levels, like glucose and lactate, in near real time. By carefully regulating growth, nutrients, and waste, the upstream process transforms a static cell culture into a highly engineered, continuously operating engine that outputs a perpetual fluid feed. That highly efficient upstream engine outputs a massive, continuous volume of fluid. When this hits a traditional step-by-step downstream purification process, it immediately creates a bottleneck. This diagram illustrates the sequential steps of downstream purification. Focusing on the first step, affinity capture, we must precisely manage parameters including load ratio, cycle time, and elution volume. To process a continuous feed without stopping, engineers use multi-column chromatography. Instead of relying on one massive column, a cyclical loop of 4 to 8 smaller columns continuously rotates. As one column finishes loading, the flow diverts to the next, while the first column moves through its washing, eluting, and regenerating phases. This multi-column approach increases process efficiency by up to 75%. It maximizes binding capacity and requires far less space. The continuous rotation of these columns successfully converts overwhelming upstream volume into manageable throughput, clearing the initial downstream bottleneck. But the fluid immediately hits a second roadblock.
Viral inactivation historically requires holding the eluate in a static batch tank for an extended period, abruptly halting the continuous momentum. Returning to the downstream purification chart, the second chevron outlines the parameters for viral inactivation.
We have to hit a specific low pH target and maintain that precise acidity over a required hold time. Rather than filling a vat and waiting, the system utilizes a coiled flow inverter. The eluate travels continuously through a specifically measured length of coiled tubing. The distance and flow rate dictate exactly how long the fluid remains inside, matching the necessary hold time without stopping. To execute this on the fly, automated inline titration injects acid directly into the moving stream. This drops the pH to between 3.4 and 3.6 instantly and maintains it throughout the transit. From there, the stream enters the polishing phase. Continuous downstream processing heavily favors a 3-column overall setup, the initial capture column followed by 2 flow-through columns. Linking an anion exchange flow-through column directly to a cation exchange flow-through column provides rapid, high-capacity clearance. As the product passes straight through, the resins strip away high molecular weight aggregates and residual host cell proteins. By engineering inline pH adjustments and utilizing flow-through chromatography, the system achieves strict viral safety and product purity, while the fluid remains in constant motion. The final physical obstacle in the downstream process is continuous viral filtration. The ultra-fine filters required to screen out microscopic pathogens easily foul and clog over extended, multi-day production runs. In the final 2 chevrons of our flowchart, we see the parameters for viral filtration and the ultrafiltration of the bulk drug substance. Both steps rely heavily on strictly managing feed flow rates and specific pressure tolerances. Since a single filter cannot survive a continuous run, the pipeline relies on automated multi-pack filter systems. When sensors detect a pressure drop, indicating a fouled filter, robotic valves automatically divert the fluid path to a fresh filter pack. Once bypassed, the used filter is taken offline and subjected to structural integrity testing. This ensures no viral leakage occurred during its lifespan, guaranteeing product safety without interrupting the live feed. Despite these mechanical solutions, a crucial technological gap remains in the process analytical technology network. True real-time, inline testing for complex impurities like host cell DNA, residual proteins, and leached protein A simply does not exist yet. Because these real-time analytical blind spots persist, the system relies entirely on the robustness of the upstream quality by design parameters and aggressive mechanical controls in the intermediate stages. These strict architectural rules are the only way to guarantee regulatory compliance and end product safety. This end-to-end continuous model operates successfully for monoclonal antibodies, but it also provides the structural scaffold for the next generation of therapeutics, specifically viral vector gene therapies. Currently, the manufacturing of adeno-associated viruses, or AAVs, relies heavily on unscalable batch methods. Purification is often bottlenecked by tedious steps like density gradient ultracentrifugation. This technical flowchart proposes a framework for AAV vector purification, applying continuous downstream logic from the initial cell harvest at the top all the way down to final formulation. The most difficult separation challenge in this process is isolating non-functional empty viral capsids from the therapeutically active fully packaged capsids. Looking at the intermediate step, this model utilizes anion exchange chromatography resins. Because empty and full capsids share similar sizes and shapes, the anion exchange resin separates them continuously based on minute differences in their isoelectric points, typically 6.3 for empty capsids and 5.9 for full ones. This precise chemical separation replaces the physical batch ultracentrifuge. Adapting continuous chromatographic logic to viral vectors allows production to scale up significantly. This engineering leap pulls gene therapies out of low-volume clinical labs and prepares them for the global market. Executing the transition to continuous bioprocessing demands massive capital expenditure, highly specialized software, and rigorous systems engineering. But the effort yields a highly efficient system. By converging multi-column fluid dynamics, PAT sensory networks, and QBD engineering, the industry eliminates the manual limitations of batch production. Facilities can reduce their physical footprint, lower their operating cost of goods,
and maintain absolute mathematically sound consistency in their final product quality. Architecting the continuous bioprocess provides the essential infrastructure required to meet surging global market demand and manufacture the next generation of medicine.